Hello to our friends joining us via recording. We are getting ready for exam number 3. Exam number 3 covers topics about the process of muscle contraction, the way that neurons work, and the functions of the parts of the brain. So to help us out today, we did some group work with classmates to remind us of some of the most important topics in this unit. So let's start here with working on the different types of ion channels that help us with the process of muscle contraction. Remember with ion channels that pretty much every type of channel in the brain or on our neurons and our muscles are gated channels. We said before we did group work that gated channels are channels that aren't always open. We have to use particular types of gate or keys to get these gates to open up for us. When we talk about a chemically gated channel, what opens up a chemically gated channel? How does this kind get open? Yeah, so broadly speaking, a chemically gated channel opens up with some kind of chemical. In this unit, when we're talking about muscles and neurons, the kind of chemical that we're looking for are things called neurotransmitters. So neurotransmitters are the messages that a neuron uses to talk to its neighbor, or in this case, that a neuron uses to talk to a muscle cell. There is one particular neurotransmitter that neurons use to talk to muscle cells, and that's this one down here at the bottom. Acetylcholine is a neurotransmitter. It's the only neurotransmitter that a neuron releases to talk to a muscle cell. Since I'm using acetylcholine, a neurotransmitter, to open up a channel on my muscle cell, what kind of gate would we say? Not the ion type, but just the kind of gate. What kind of gate is on this particular channel if acetylcholine opens it? What did we just say that was? Yep, that's going to be my chemically gated channel. In particular, the type of ion that I'm going to rush into my cell when I open up this kind of channel is an ion that's going to make my membrane charge get more positive. I want to excite my cell. Which of these ions that we see right here do I let into my cell when I get acetylcholine? Who comes inside? Yeah, sodium comes inside. So it's a chemically gated sodium channel. The chemically gated sodium channel. This is found at the neuromuscular junction where a neuron talks to a muscle cell. On the membrane of the muscle cell, we have chemically gated sodium channels. And those chemically gated sodium channels open up. In comes the sodium and my membrane charge goes up. That's going to allow me to generate something called an action potential. And if you remember from our discussions of action potentials in muscles and in neurons, think about an action potential just as a place where the membrane charge changes and it moves places. This action part right here means it's moving. Potential means it's a membrane charge. I need to make my membrane charge go up, and then I need it to come back down for me to have an action potential. To start my action potential, I am responding to the fact that sodium came into my cell. When sodium rushed into my cell, that made my membrane charge change. When my membrane charge changes and that opens up my gate, which kind of gate is it that's opening when it has to do with membrane charge? Which kind of gate opens with those charges? Yeah, voltage gates. Voltage gated channels are the types of channels that when you change the membrane charge, they open up. When we're talking about action potentials on neurons or on muscle cells, there's two types of ions that are involved. The first kind of ion that is involved is the kind of ion that makes the membrane charge more positive. So it's the same thing as we were talking about with acetylcholine. The first kind of ion channel that opens up for an action potential is a voltage-gated sodium channel to make my membrane charge go up. When my membrane charge gets completely up, though, my muscle cell realizes I should not be up here. I need to go back down. It's going to use a voltage-gated potassium channel to help it go back down. The two channels responsible for action potentials, for making the charge go up and the charge go down, are voltage-gated sodium channels and voltage-gated potassium channels. The last kind of channel that's involved in the process of muscle contraction is a type of ion channel that opens when it's pressed on, or when something pushes against it. Which kind of gates open when they're pressed open? When I push on it. Yeah, long name, right? The kind that opens in response to it, it's called mechanical energy, which is like pushing energy. The kind of channel that opens because of that is a mechanically gated channel. So when we're talking about the end of excitation-contraction coupling, the way that that process happens is we push open a type of channel that's found on the sarcoplasmic reticulum. When I press it open, it spits out calcium. I have mechanically gated calcium channels. They're pressed open when some voltage-sensitive proteins, meaning proteins that are listening for the action potential, when the action potential comes down that T-tubule, it changes the shape of these voltage-sensitive proteins. They push on mechanically gated calcium channels and spit out calcium. Calcium is what I need to start the cross-bridge cycle. So here are our four main types of channels involved in muscle contraction. Let's take this mechanically gated calcium channel here and apply it to the next activity that we did with our classmates here. When I spit out that calcium from the sarcoplasmic reticulum, who does the calcium attach to? Which protein does calcium actually go down to and attach to? Yeah, the protein that calcium directly attaches to is called troponin. Remember that we said troponin was our pushpin protein. It's our protein that holds somebody else in place. Who does troponin hold in place? Which protein is troponin helping out with? Yeah, so it's helping with that big long one called tropomyosin. The job of tropomyosin is to cover the myosin binding sites. You can use its name to help you realize that. Tropomyosin covers the myosin binding sites. So let's add that here. Tropomyosin. So here's my pushpin protein that normally holds that wet spaghetti noodle, tropomyosin, in place. When I release calcium, the pushpin pops out and the spaghetti noodle slides out of place. Now I can form what's called the cross bridge, where myosin and actin can interact with each other. When myosin and actin interact with each other, one of them is going to move, or one of them is going to slide. It's going to be pulled past the other one. Do we remember, was it myosin or actin that actually moves during muscle contraction? Yeah, exactly. Actin is the protein that actually moves. It moves because myosin that attached to it changes its shape. Myosin changes its shape because at one point it was attached to ATP, but then it breaks down that ATP, and when it breaks down that ATP, that changes its shape and allows it to pull along actin. So the last protein that we're labeling here is myosin, the protein that breaks down ATP and forms a thing called the cross bridge when it interacts with actin. So that's the sum of the group work that we did related to muscles. Let's switch topics now and talk about neurons. When we talk about neurons, we said this before we split into groups, that excitatory neurotransmitters generate EPSPs. EPSPs. Now, that EPSP word means excitatory postsynaptic potential. Again, we can see that word potential. It still means membrane charge. This excitatory part right here, does that mean that my membrane charge goes up or goes down if I have an excitatory change? Yeah, excitatory changes, excitatory equals positive, a positive change. We would say that this is excitatory, or it's a positive change, because remember that I have to get to threshold. I have to get just positive enough to be able to completely depolarize my cell. So an excitatory neurotransmitter is a neurotransmitter that's going to make the membrane charge in my postsynaptic neuron go positive. So when we look at our various types of ion channels that are over here, we get to come back to Dr. Ollis's favorite thing ever about cells. For my friends who are our regulars here in office hours, what am I about to draw for us here to help us answer this question? Yep, here it comes. That salty banana, my favorite. Okay, if you're new to office hours, hopefully you have at least seen some of our previous videos where we talk about the salty banana. This is the best way for you to remember what I have a lot of inside versus what I have a lot of outside. So let's start on the inside. We've got a banana. A banana is really high in what? What's on the inside of this banana? Yep, potassium, K+. Lots of potassium inside my banana. And it's a salty banana. On the outside, I have the stuff that makes up salt. The two things that make up salt are, yep, got a whole lot of sodium and a whole lot of chloride. Absolutely. Okay, here's my salty banana. It's my cell. I've got a lot of potassium inside, a lot of sodium and chloride on the outside. When I open up channels for any of these ions, they are always going to move down their concentration gradient. They're always going from where there's a lot of them to where there's a little of them. So when I open up a channel for sodium, is sodium going to go into the cell or out of the cell? Which way is sodium going? Yeah, sodium, we have a whole lot on the outside. Sodium's going to go inside. Absolutely. Same thing with chloride, right? Chloride's going to come inside too, because there's a lot of it on the outside. Potassium, there's already a bunch inside. So potassium is actually going to go out of my cell. It's going to leave my cell. Okay, so the first thing we've done is identified which way things move. Now we need to figure out what effect that has on the membrane charge. Sodium has a positive charge. When sodium comes inside, does it make the inside more positive or more negative when sodium comes inside? Yeah, sodium has a positive charge. So when I bring in a bunch of positives, that's going to make the inside more positive. So sodium channels, we would say, are excitatory. They're making the membrane charge go up. Chloride is an ion with a negative charge. When I let chloride into the cell, what happens to my membrane charge? Does it go up or does it go down? Yeah, chloride is going to make my membrane charge more negative. It's going to go down because chloride has a negative charge. We would say that chloride coming in would generate an inhibitory postsynaptic potential. It's going to go more negative. That leaves us with potassium. Potassium has a positive charge. Potassium leaves the cell though. When potassium with its positive charge leaves the cell, it's kind of like we're doing subtraction. So when potassium leaves, it's another way for the inside to become more negative. Inside becomes more negative. So when we're talking about an excitatory neurotransmitter, which of these ions that we mentioned here, sodium, chloride, or potassium, which of these ions generates an EPSP, an excitatory postsynaptic potential? Exactly. Some of us are pointing out that the only one that made my membrane charge go up was sodium. So for an excitatory neurotransmitter, something that makes a positive change in charge, the only way I can do that is opening a chemically gated sodium channel. Now, I'm going to stay on this page and we're going to change the question to what the other one was because I've got my pretty salty banana here. Let's say that instead of excitatory, we're talking about an inhibitory neurotransmitter. Now we're making an IPSP. What happens to the membrane charge with an IPSP? What happens to my membrane charge? Yeah, it's going to go down, right? So it becomes more negative. If I spit out a neurotransmitter that's an inhibitory neurotransmitter, that inhibitory neurotransmitter would make my membrane charge more negative. Which types of ion channels could I open to make my membrane charge more negative, to make an IPSP? Exactly. A lot of us are chiming in that there are actually two types of channels I could open to make my membrane charge more negative. The first way to make it more negative is to bring more negatives inside. That's what happens when I do a chloride channel. The second way is to open up a potassium channel. Potassium with its positives will leave. So when I take those positives away, that makes the inside more negative as well. So big picture, important idea for us as we're getting ready for exam number 3. We've got to know that salty banana. We got to know where there's a lot on the inside versus on the outside. Which way are things going to move and what's going on with their charges. So for my original question, the excitatory neurotransmitter, the correct answer was sodium channels only. If we change it to an inhibitory neurotransmitter, we just talked about how potassium and chloride, both of these types of ion channels would make the membrane charge go down, would make it more negative. Tell me in the chat, thumbs up, thumbs down, how do we feel about this particular question? Okay. So several of us are feeling okay about it. Perfect. All right, so let's talk about the last thing then that I had us do with our groups. The last thing I had us do was to remember what some of the parts of the brain do. So some of the things that I asked you about were normal functions. Some of the things were abnormal functions. Let's start with the normal functions. So the first thing that is the normal function of something is the part of your brain that makes you feel scared. Which part of your brain makes you feel scared? Yeah, so it's a weird spelled one, right? It's a thing called the amygdala, or technically our pictures have it labeled the let's see if I can spell it right. Amygdaloid body. Either of those, exactly the same thing. We'll give you both on the exam, but it's the same thing. So the part of your brain that makes you feel scared is the amygdaloid body. The amygdaloid body, or the amygdala, this works together with things like the olfactory bulb and the hippocampus to help you form what's called your emotional brain. What's the name of your emotional brain? What did we say that name for that overall is? Yeah, exactly. The emotional brain, I guess I should put that in quotes because it's not the technical science name for it. The emotional brain is the structures that are found in the limbic system. So the limbic system is where you go with more emotional responses to things like fear. That's from the amygdala. Things like remembering how a situation makes you feel. That's the hippocampus. The olfactory bulb helps you to have an emotional response to what you remember. So the limbic system, all about those emotions. When we're talking about something like controlling the movement of your fingers, and in particular, your fingers have skeletal muscle, which part of the brain has skeletal muscle? Yeah, so we're controlling it with the frontal lobe. I asked that question really terribly. I apologize. There's a part of the frontal lobe that specifically controls skeletal muscles. Yeah, so and I heard this discussion in several of my groups. There's kind of two ways we could answer this question. The first way we could answer it is to name the specific place in the brain where we control skeletal muscles, and that's something called the pre-central gyrus. That's the specific bump found in front of the central sulcus that helps us to control skeletal muscles. More specifically, in that bump, as several of us have identified in the chat, we have that thing called the motor homunculus. The motor homunculus. And remember, this homunculus word just means it's a map of the body. So the specific part of your brain that's controlling skeletal muscles, whether it's in your fingers, in your toes, in your arm, wherever it is, that's coming from the map called the motor homunculus that we find in the pre-central gyrus, that bump that's at the very end of the frontal lobe. By the way, what's going on in that structure called the post-central gyrus? The post-central gyrus. What's up with that one? What's the function of that bump? Yeah, so that's going to be the one that has the sensory homunculus, the map of where you send all of your sensory information. So if I had changed this question, instead of controlling the skeletal muscles in your fingers, if instead it said that it was receiving sensory information from your fingers, that would be the job of the post-central gyrus. Motor control, pre-central gyrus. Sensory stuff, post-central gyrus. Along the lines here of movement, there's a specific part of your brain that helps you to not do movements that you don't want to. Which part of your brain prevents you from doing unwanted movements? Or it stops unwanted movements? Yeah, so the part of the brain that blocks unwanted movements are the basal nuclei. Remember, the special thing about these is that they are made out of gray matter, but they're found in the very middle of the brain. Most of the gray matter of the brain is on the outside, but these are gray matter in the very inside, and their job is to prevent unwanted movements. So remember when we talked about things like Tourette syndrome or Parkinson's, where there's tics, which is a brief, fast movement, or things like a tremor that's a repeated movement that you can't stop. Those are problems with the basal nuclei where they're not blocking those movements. Miriam has a question. Go for it.
Sorry, it might not be related to the basal nuclei, but one question just wanted to ask regarding gray matter and white matter. Is the gyri and sulci consider a gray matter?
Yeah, so the structures of the gyrus and the sulcus, so that's basically just the raised structures and then the indentations between them, they're completely covered in gray matter. So yes, that would be where in the brain we see the gray matter is in those bumps and those indentations that are on the outside. All of the very middle of the brain is where we'd find that white matter, except for these guys right here, the basal nuclei. Those are gray matter in the very middle.
Okay,
Most of
thank
that gray
you.
matter, though, yeah, is arranged in those things called gyri and sulci, the bumps and the indentations. Yeah.
Thank you.
Yeah, you're welcome. Okay, so we said controlling skeletal muscles comes from the pre-central gyrus. Stopping unwanted movements of those skeletal muscles, that's the basal nuclei. There's one other part of the brain, a part that's really easily inhibited by alcohol, that helps your movements not to look stupid or not to look silly. Which part of your brain is easily affected by alcohol? Yeah, that part over here is the cerebellum. The cerebellum, easily influenced by alcohol. So when we were talking about the functions of the cerebellum, basically just think about what drunk people can't do well. That's what the cerebellum does for us. So the job of the cerebellum is to smooth out your movements, to make them look correct. Um, I I don't know, I mentioned it in class. Did anyone happen to read any of those articles about the people born without a cerebellum? I'd mentioned we got a couple of current event articles that were about people without a cerebellum. So it does more than just the movements. It does some other things as well. Yeah, go for it, Hannah.
Okay, now that you mentioned that article, I read that one about the lady who was born
Mm-hmm.
without a cerebellum, and she didn't figure it out until she was like older. And I
Mm-hmm.
was so confused on how she didn't figure that out until then. You know, for her
Yeah.
parents who have watched her since she was a child. You know what I
Uh-huh.
mean?
Yeah. Well, and I think a lot of it comes down to attributing some of those deficits to other things. Because it's so rare that someone would not have a cerebellum. If they're clumsy, if they're slow to develop, that's not going to be your doctor's first assumption. Oh, you probably don't have a cerebellum. And I think too, with modern brain scan technology, the stuff that we do in utero, we'd be much more likely to catch that now than perhaps they were back when her mother was pregnant with her. So I think we would know about it now. I'm not sure why they didn't know about it earlier. That's actually a really good question. But yeah, it was fascinating that she could live her whole life without a cerebellum. It doesn't make things work absolutely perfectly, but it is possible to live without it. So, all right. So we talked about being intoxicated, how that's a cerebellum problem. Which part of the brain was a problem with jet lag, or just with feeling exhausted all the time? I'm about exhausted all the time. That's where we're at. Let's be real, right? Yep. Jet lag is a problem with pineal gland. Pineal gland. That's the part of your brain that makes the hormone melatonin. So melatonin made by the pineal gland when you're tired. It's because you have higher melatonin levels. Yeah, Karina mentioned another cup of coffee. I'd seen a really funny meme a while back that said, "I'm not an early bird. I'm not a night owl. I'm just a permanently exhausted pigeon." And so that's about where I'm at. I'm going to blame my pineal gland for being a permanently exhausted pigeon. Not a night owl, not a early bird, just a permanently exhausted pigeon. All right, last thing that we mentioned here in our group work that we worked on was talking about the parts of the brain that are involved in breathing. There were two specific parts that we listed as helping with breathing. What were the two specific parts for breathing? Hannah's the only one that's voted. There we go. Getting a few more. Okay. Yep. Our votes are correct. The two parts of the brain that help us with breathing are the pons and the medulla oblongata. The pons and the medulla oblongata. Both of these are found in a part of the brain called the brain stem. So the brain stem is responsible for keeping you alive. We have what's called the pontine respiratory center and the medullary rhythmicity center. Those two things work together to dictate your breathing rate. But we also have other things that the brainstem does for us. And I heard several of my groups talking about this. So some of your reflexes, things like your heart rate, there's a lot of different things that come from the brainstem to keep you alive. It's more basic to keep you alive. So compared to some of these other functions that we talked about over here, when we talk about the brainstem, remember that's your more basic brain. Your bigger picture brain, your what makes you human brain, some of these other parts of the brain. Before we move on, do we have any questions about what we've discussed so far before we switch gears? Yeah, go for it, Miriam.
Before we leave the parts of the brain, we had a question in the guided lesson regarding a person that is unconscious and at the same time he is breathing on his own. He doesn't need a ventilator. And I get the part which there were two questions talking about which parts of the body of the brain actually working and which parts that are not working. I got the part that where I know where which part that is not working, but I'm kind of confused and specifically what parts of the brain at this moment that we should mention that it is actually working.
Yeah, so that's a great question. I want to put that question to the chat. We talked a little bit about it in one of our classes last week. What were some of the things that we said would still be working in that particular patient? Do you remember? Yeah, so Christina kind of gave us the big picture of it. Really the parts in this patient, because this patient can breathe on his own, he still has some of these more basic areas of his brain working. So the pons and the medulla oblongata that work together to help him with breathing rate. The fact that it says he's not on a ventilator, he's breathing on his own. These things would still be working. He probably still has some reflexive functions that are working too. So coughing, vomiting reflexes, those are kind of things that come from the brainstem as well. Jacqueline, we didn't really talk about the midbrain in lecture, but for your reference in lab, yes, the other part of the brainstem is the midbrain. So the midbrain, I'll just put a little note about it. It doesn't help with the process of breathing or your heart rate, but the midbrain does have a lot of reflex centers in it. Reflex centers for your vision and for your hearing. So those would be part of the brainstem as well. So the brainstem is probably working. Oh yeah, they're not the same thing, Jacqueline. It's easy to mix them up. They're not exactly the same. The midbrain is part of the brainstem. Some of the other parts of the brain, I see Gloria chiming in. We kind of toyed back and forth with what other parts are actually working. You could probably make a case for some of those other parts like Gloria listed. There's one big part though that we could say definitely is not working. What's the part of the brain when a person's in a coma? Which part is definitely not working? Yeah, so the part that's definitely not working is the part of the brain called RAS. That's an abbreviation for the reticular activating system. The reticular activating system. Its job is to keep you conscious. If you're in a coma, you are not conscious. Therefore, obviously, the reticular activating system is not working because its job is to keep you conscious. And again, we could debate about some of those other parts of the brain, whether or not they're working. The most straightforward answers, though, for what is and isn't working, the brain stem, some of those basic structures, is working. The reticular activating system is not. We could then toss into some of the stuff that's not working. Since you're not conscious, you're not thinking, you're not moving your body. So all of those lobes of the cerebrum most likely are not working either, because that was the job of RAS, to keep them working. If we don't send them the pings to wake them up, they're probably not working either. Kai, to your question about the pineal gland, we debated that one a little bit. My gut instinct would be that probably the pineal gland is not functioning correctly, just because when you're in a coma and your eyes are closed, you're never really perceiving the difference between light and dark. And that's the way that the pineal gland knows to spit out melatonin at the right time. So my inclination would be to say that the pineal gland probably is not working correctly, but it's hard to say for sure. I know we can say for sure that RAS is not working. I know we can say for sure that the pons and the medulla are working. So those are the biggest, most important ones for us to know on that. But yeah, great question to mention that one. All right, I'm looking at my list here of the topics we wanted to cover. Let's start with this. This help me out in the chat. This is the lesson number 9 packet. What page in the lesson number 9 packet are we on with this one? What page is this on? Okay, I'm hearing maybe page 10. Yeah, okay. So what this picture shows you is something that sounds a lot scarier than it actually is. I promise it's not as scary as its name sounds. This picture shows us what's called excitation-contraction coupling. Excitation-contraction coupling means how do I get from exciting a cell, meaning changing its plasma membrane charge, to getting that cell to contract. Excitation-contraction coupling. A neuron talks to a muscle cell and the muscle cell contracts. That's all that excitation-contraction coupling means. The way that this process works is a neuron spits out neurotransmitters. Remember, we talked about those neurotransmitters, right? They're called acetylcholine. ACH is the abbreviation. We spit out those neurotransmitters. Those neurotransmitters go down and attach to chemically gated sodium channels. The chemically gated sodium channels make the membrane here at the neuromuscular junction get positive. We've just started to generate what's called an action potential, a moving change in membrane charge. So let's say that this part right here that's showing up close on my plasma membrane, let's say that's right about here on my previous picture. So what happens is this chemically gated sodium channel opens, sodium rushes inside the cell and makes the inside of the membrane over here positive. As this part of the membrane gets positive from all that sodium that came inside at this channel, that causes the voltage gated sodium channels that live over here to open up. And when they open up, more positive sodiums come inside, and that causes these voltage gated sodium channels to open. And now more positive sodiums rush inside. And that causes these voltage gated sodium channels to open, and sodium rushes inside. This whole process of rushing in sodium to make the inside of the cell positive, that's going to get a message all the way across the outside of a muscle fiber. Remember that in muscles, their membrane is called the sarcolemma. Sarco means muscle. We don't, though, just want to freak out the outside of a muscle fiber. We need to change the membrane charge all the way down inside the muscle fiber because we need all of its proteins to contract. So remember that we have these structures called T-tubules. These are places where I folded in the plasma membrane of my cell. I fold in the plasma membrane, and now the sodium that came inside the cell right here causes these voltage-gated sodium channels to open up, and they bring in some sodium. Well, when this part of my membrane gets positive, notice that I have a voltage-sensitive tubule protein. We talked about this way back at the beginning of our time today. A voltage-sensitive tubule protein. This is a protein that lives inside the T-tubule. When the membrane charge gets positive, this changes its shape and presses on my mechanically gated calcium channels. My mechanically gated calcium channels are the gate that decide whether or not calcium gets to leave the sarcoplasmic reticulum. So when the T-tubule, that place where the membrane folded, when this changes its charge, it changes the shape of my protein embedded in the T-tubule. That pushes open the calcium gate, and calcium leaves the sarcoplasmic reticulum. When calcium leaves the sarcoplasmic reticulum, it goes down and attaches to that push pin. And when it attaches to that push pin, it pops out of place, and this spaghetti noodle, tropomyosin, moves out of place as well. That allows myosin to attach to actin and actually move actin to do the process of muscle contraction. Excitation-Contraction Coupling. We took a change in membrane charge that started right here, where a neurotransmitter attached to an ion channel. We changed the membrane charge everywhere, including inside these little T-tubules. That allowed us to spit out calcium inside the muscle cell, which allowed us to do the process of contraction. Help me out in the chat. What questions do we still have? Yeah, so Ariel asked a good one. This picture right here would absolutely be where we see the cross-bridge cycle starting. Because remember, the cross-bridge cycle, where myosin attaches to actin and then pulls on actin and then attaches to ATP, all that stuff, that can't happen until calcium gets spit out. So once I spit out calcium, then I can do the cross-bridge cycle. So I'll put a little note here. Cross-bridge cycle. That's where myosin and actin interact with each other and actually make movement. So yep, that's the third part of this process. Yeah, and so Nicole asked a good follow-up question too. She asked about the voltage-gated potassium channels. When we talk about voltage-gated potassium channels, when they open, what do they do to the charge on the plasma membrane of a cell? When voltage-gated potassium channels open, what do they do to membrane charge? Yeah, they're doing what's called repolarizing. So here, I'm going to draw us a little graph here. When we're talking about the way that muscle cells work, either they're depolarizing their membrane or repolarizing. Oops, my pointer died there. Repolarizing their membrane. The way that we depolarize the membrane, make my membrane charge go up, is by letting in sodium. The way I make it go back down is by spitting out potassium. Because remember, potassium has a positive charge. So we spit out potassium, we do subtraction, basically, to get our membrane charge back to normal. Ooh, that's a good review for us. What is the resting membrane potential, the normal charge on a muscle cell? What's a muscle cell resting membrane potential? Ooh, we're debating. Is it -70 or -90? Which one is it for a muscle? Yeah, so here's where it's a little tricky. Muscle cells have a resting membrane potential of -90. They are more negative than neurons. Neuron resting membrane potential is -70. When you are taking your exam today or tomorrow, please check the wording on your question. Make sure you're not telling me the charge on a neuron if your question asks you about a muscle cell. Or make sure you're not telling me about a muscle cell if your question asks about a neuron. It should be very obvious. I try not to be tricky, but I just want to make sure you're answering the right question. So, resting membrane potential on a muscle cell, -90. I use, like you told me, I use sodium rushing into the cell to depolarize it. I use potassium rushing out of the cell to repolarize it. Ariel asked about a thing that we talked about in the neuron chapter called threshold. So the thing with muscles is muscles are what we might call all or nothing. If you start depolarizing their membrane at all, they will always do this entire process. That's not the way it works in neurons. In neurons, remember, they do that process called summation, where you can give me an excitatory message or an inhibitory message, and I'll mix it all together, see what I get. Yeah, in a muscle fiber that starts at -90, there's none of that summation business. Either the muscle is getting a message that says it should contract, or it's not getting any messages at all. So muscles don't have threshold. Muscles are just at -90. If I go positive at all, that's going to open up my voltage-gated sodium channels. They're very sensitive. So all that to say, we don't do summation in a muscle cell. It's all or nothing. If you start going positive, we're going all the way positive. Yeah, all the way up, like Ariel mentioned, all the way up at the top to +30. +30. And when we're up here at +30, our cell realizes, oh my gosh, I'm supposed to be -90. I'm +30. This is not good. I'm going to open up my voltage-gated potassium channels so that I can get my membrane charge back to -90. Get me back to where I belong. What other questions do we have about this stuff? Yeah, go for it, Miriam.
So, in this graph, one thing that I don't really get, which is the part of when the voltage-sensitive tubule protein channel opens.
So they are going to open around this +30. The clarifying on that, and it's kind of a good clarification to make here, we don't see these voltage-sensitive tubule proteins anywhere except inside the T-tubules. So except where we're right next to the sarcoplasmic reticulum, you're not going to see the voltage-sensitive proteins. So these proteins are only found in the T-tubules. They're going to press on the proteins in the sarcoplasmic reticulum. So the entire membrane is all going to go up to +30, and then it'll all work its way back down. If we happen to change this part of the membrane to +30, that's going to change the shape of the voltage-sensitive tubule protein. When I change that shape, that's going to press on these channels. It's kind of like, if we relate this to neurons, how, remember, in neurons, we have voltage-gated calcium channels. In neurons, they're actually voltage-gated. Remember that in muscle cells, they're not voltage-gated. These are mechanically gated. In neurons, we don't have that extra tubule protein to press on us to open us up. We open up on our own, a voltage-gated calcium channel. But this particular type of channel is only seen in the part of the neuron where we actually spit out neurotransmitters, because that's the job of calcium, to actually spit out neurotransmitters, make them spit out. Which part of a neuron's membrane has voltage-gated calcium channels? Where are those channels found on a neuron? Does anyone remember? Yeah, those channels are found on what's called the axon terminals. The voltage-gated calcium channels are found at the end of the line, the place where a neuron spits out a message to its neighbor. Here, let's do our corollary here with our muscle cells. Mechanically gated calcium channels, type those. Mechanically gated calcium channels, they're found on the, I'm going to run out of space, sarcoplasmic reticulum. They're found on the sarcoplasmic reticulum. So where the sarcoplasmic reticulum is right next to the T-tubule, the place where the membrane got folded in, that's where they'll interact with each other. But the rest of the sarcolemma is not going to have these voltage sensitive proteins, and it's not going to have those mechanically gated calcium channels. These are only found on the sarcoplasmic reticulum, and these sensitive proteins are only found on the T-tubules. Does that clear it up for you, Miriam, or do you still have a question with it? Perfect. Any other questions? Yeah, go for it, Jacqueline. So the question is for my friends watching the video. The question is, do we need to know about the voltage-gated potassium channels for the sake of lesson number 9? What I would say is they do the same thing in lesson number 9 that they do in lesson number 10. So if you know that voltage-gated potassium channels always help to repolarize the membrane charge, it's the same thing for muscle cells as it is for neurons. So know them in the context of an action potential. This is how I get things back to normal. That's all you need to know about them, that they help us get it back to normal. Yeah. Okay, let's talk. Let me pull up my picture here. One of the things that we wanted to talk about was those disorders. So let's dive in right here. We said this is where the cross-bridge cycle happens. Let's talk about the cross-bridge cycle briefly as a segue into rigor mortis for us. Excuse me. The cross-bridge cycle has 4 steps. Remember that we defined, let's type this out here. The cross-bridge cycle. A cross-bridge is just where myosin and actin attach. That's what a cross-bridge is. The cross-bridge cycle is the way that I attach myosin and actin to each other and the way that I get actin to move. An important part of the cross-bridge cycle, they're all important parts, right? But several important things happen in the cross-bridge cycle. First thing that happens is myosin and actin attach to each other. If the myosin binding sites, so these circles here on my blueberries, if these myosin binding sites on actin are available, myosin will always go up and attach to it. It's just the way that things work in the body. If this spot is open, myosin is going to attach to it. So when we talked about excitation-contraction coupling, how we spit out calcium, the goal of spitting out calcium was to get us right here, where calcium attaches to my pushpins troponin and slides tropomyosin out of place. If I don't have calcium, none of this cross-bridge cycle stuff would happen. None of this is going to happen because tropomyosin would be covering those myosin binding sites. So for me to be able to do the cross-bridge cycle, I have to spit out calcium first. That makes it possible. If the myosin binding sites are available, the first step of the cross-bridge cycle is myosin attaches to actin. That's called cross-bridge formation. when myosin attaches to actin. The second thing that happens is actin slides. The reason that actin slides in this thing called the power stroke or the working stroke, the reason that actin slides is because myosin tosses out its trash. So ADP and inorganic phosphate, these two little things right here, this is what's left over when myosin chops up ATP, which is its energy source. I hold on to it for a little while after I've chopped it up. So I chopped it up over here. It stays attached to me for a while, but then I realize it's kind of a drag. So I spit it out. When I spit out ADP and phosphate, that allows my myosin head to bend over. When it bends over, it pulls actin with it. So in the power stroke, actin slides toward the middle of the sarcomere. It actually moves for me. At the end of this step, myosin and actin are attached to each other. The way that I get myosin and actin to let go of each other is I bring over a new molecule of ATP. When ATP, this high energy molecule, comes and attaches to myosin, myosin realizes, actually, I don't need to hold on to actin. I've got something way cooler. So myosin is attached to ATP and it lets go of, or it detaches from actin. The last stage of the cross-bridge cycle, though, is myosin chops up this energy molecule, ATP, helping it to go back into what's called the cocked position, where it's ready to attach to actin again. So to go from being bent over in this position we see here, back to the cocked position where we're upright, we have to break down, or it's called hydrolyze ATP. When we hydrolyze ATP, we're back in the right shape to be able to attach to actin and do this whole cycle over again. So the cross-bridge cycle, all about getting myosin and actin attached to each other and sliding past one another. Ariel asked the question, if the cross-bridge formation step is where ATP is released. ATP is actually never released. What I do with ATP is first I attach to it, then I chop it up. So I took my ATP, here, let's do some review here. ATP equals adenosine triphosphate. When I chop it up, it becomes ADP, which is adenosine diphosphate. So over here, it's ATP. It's got a lot of energy. Then I chop off one of its phosphates. It goes from being ATP to ADP. I'm still holding on to it, and I'll actually keep holding on to it until I get back to the power stroke. So when I let go of what's left of my ATP, which is the ADP, when I let go of it, that's the power stroke. I'll hold on to it until that point. Once I let go of it, that changes the shape of myosin and allows me to pull actin past me. Now, the cross-bridge cycle relates to rigor mortis. That's the stiffening of the body after death. Which of the stages of the cross-bridge cycle can we not do in rigor mortis? Which of these stages can't happen after you've died? Exactly. Yep, we're all honing in. The stage of the cross-bridge cycle that can't happen after you die is cross-bridge detachment. Cross-bridge detachment is when myosin lets go of actin. And the reason that myosin lets go of actin is because ATP came and attached to myosin. When a person dies, they stop making ATP. So myosin and actin get stuck. We get stuck here at the end of the power stroke. They're attached to each other, and there's nothing to tell myosin to let go of actin. So the stage of the cross-bridge cycle that we can't get past when when we die that leads to rigor mortis, we can't get here. We can't detach myosin and actin. That makes things really stiff. Rigor mortis. Yeah, go for it, Ariel.
Okay, so on the review assignment, they were talking about that during the blank, the energy that was stored in ATP is released, allowing myosin to change its shape. So that's why I asked that question. So I put detachment, but I wasn't sure.
Yeah, so when we talk about releasing the energy that's stored inside ATP, that is what we do right here in this last stage, where we do ATP hydrolysis. So I chop up ATP and I use the energy that was inside that molecule to make my myosin head stand back up. So before I've chopped up ATP, myosin is bent over, it's in the low energy state. Once I chop up ATP, it turns into ADP, but it also gives me the energy to change my shape to be standing upright. So the stage where I release that energy, or where I can use that energy to change my shape, would be that last one, the cocking of the myosin head, getting it back into the position where it would be able to attach and do the cross bridge cycle again.
Okay, thank you.
You're welcome. That's rigor mortis. Let's bump to sarin gas. Can you help me out in the chat? Which page in your notes do you have this picture? What page is this one from? Okay, page 11. This picture shows me what happens when, what normally happens, let's put it this way, what normally happens in the body when you've sent a message for a muscle to contract and then we're done. So here's my neuron up here at the top. Here's my muscle cell down here at the bottom. So I'm up here inside the neuron. The neuron has acetylcholine. That's my neurotransmitter. The neuron spits out acetylcholine out of the neuron. That acetylcholine comes down here and attaches to, remember, our chemically gated sodium channels. So in normal processes, when things are working the way that they should, a neuron spits out acetylcholine, that activates chemically gated sodium channels. That's going to lead me to make an action potential and do the process of muscle contraction. Normally, once I've heard this message, then I'm going to use what's called acetylcholine esterase to chop up that message. I only want to hear that message once because it was only released once. So in normal circumstances, I attach acetylcholine to the chemically gated sodium channel. That chops up acetylcholine into its two pieces. These pieces by themselves I can't do anything with them. They're not going to make me contract my muscle. So I'll basically recycle the parts and build it again. In normal circumstances, that's what we do. If someone is exposed to sarin gas, what happens is acetylcholine esterase, my enzyme whose job it was to chop up that message after I've heard it, acetylcholine esterase gets inactivated. So I stop being able to chop up a message after I've heard it. What happens with sarin gas exposure is I hear my message once, it depolarizes my membrane, makes it more positive, but then that same message goes and finds another receptor on the membrane, and I respond to it again. And then I let go of that receptor, and maybe I go find another receptor, and I respond to that message over here. I just keep responding to that message over and over again. It's not because I spit out too many messages. It's that I spit out one message and heard it about 5 or 10 times before it just happened to float away. So the problem with sarin gas is acetylcholine esterase is inactivated. It can't chop up acetylcholine when I'm done responding to it, which means basically I respond to it forever. So in sarin gas, we block acetylcholinesterase. No way for us to chop up those messages. The other medical correlation that we made, aside from sarin gas, is we talked about Botox. Botox is not a problem on the muscle side. Nothing's going wrong with the muscles. What happens in Botox is I don't actually release my neurotransmitters. And you have more specific pictures in your notes that talk about exactly why we don't release neurotransmitters. But botulinum toxin, what Botox actually is, it goes inside the neuron. It chops up a protein that helps this bubble with the neurotransmitters to come down to the membrane. And it chops up a protein that's here in the membrane that kind of acted like an anchor. If I chop up those two things, the neurotransmitters get stuck inside the neuron, and I can't spit them out of the neuron. If I can't spit out those neurotransmitters, my muscle can't respond to them because it never sees them. So we kind of phrased it this way back when we were talking about things. When we talk about sarin gas and when we talk about Botox, which of these is a problem where I have too much acetylcholine at the neuromuscular junction? Which one of these I have too much acetylcholine? Yeah, too much acetylcholine, too much ACH. That's the problem with sarin gas. I have too much, not because I spit out too much. I spit out the normal amount. I have too much because I can't chop up the normal amount. It just keeps floating around forever. I can't chop it up. When we talk about Botox, what would we say about acetylcholine? Too much or too little? When someone has Botox, what's going on with their acetylcholine? Yeah, when someone has Botox, they have too little acetylcholine, or more accurately, they have no acetylcholine. So these conditions showed us the two opposite extremes, if you will. They showed us how important acetylcholine is. Acetylcholine starts the process of excitation-contraction coupling. If I don't have acetylcholine, I can't start the process at all. If I have too much acetylcholine, I never stop the process. So I need to have just the right amount. When I'm done with it, I need to chop it up and recycle it, and then I'll have normal functioning of my muscles. Sarin gas, Botox, rigor mortis. Those were our big correlations when we were talking about the muscular system. Any questions about those things before we move on? I've talked a lot today, and we haven't had nearly enough emojis. Can we have an emoji fest here for a moment? How are we feeling? What are we thinking about? Where are we at? Send me some emojis. Nice, I like it. Anything to be lucky, right, Hannah? Dragon. Don't do that tongue thing. For any Mulan fans. All right. Well, let's see. I think the next thing we'll do is we will look at this. We've moved on to lesson number 10 now. Can you tell me what page we're at on lesson number 10? Where are we at on 10? Okay, so page 12, I'm hearing. Okay. This is one of those activities that I would highly recommend if you have the ability to reprint pages, to do them multiple times to make sure you're ready for the exam. This would be an activity that would be a great one for you to redo to help you with your studying, make sure you're on top of things. The goal with this particular activity was to help us remember where I find different kinds of ion channels on a neuron. And then we coupled that with what their jobs are when we're looking at that action potential graph. So we'll start with labeling which kinds of ion channels live where. Then we'll correlate that to the graph for the way that a neuron works. When we're talking about neurons, notice that in neurons, we only have chemically gated channels and we only have voltage gated channels. Neurons don't have any of the mechanically gated channels. That was just the muscle cells. You've already told me this once today, but let's make sure we remember it. A chemically gated channel, any of my types of ions that we see here, a chemically gated channel, how do I open those again? How do chemically gated channels get opened? Yeah, exactly. I need some kind of chemical. Again, just like we saw with the muscles, when we're talking about chemically gated channels on neurons, the type of chemicals that are going to open them up are the neurotransmitters. So the chemicals are neurotransmitters. Now, when we're talking about neurons, talking to each other, we use more than one type of neurotransmitters. Remember that we just use one neurotransmitter when a neuron talks to a muscle. It always just uses acetylcholine. When neurons talk to each other, they can use a whole bunch of different neurotransmitters. Regardless, though, of which particular neurotransmitter neurons are releasing to each other, there's really just kind of one place on a neuron where they can receive neurotransmitters. Which part of a neuron is the receiving part of a neuron? Where do we have neurotransmitters? Yeah, exactly. The place on a neuron where we have neurotransmitters is the part called the dendrites. So the dendrites are all of these little branches that we see here coming off the part of the neuron called the cell body. So my dendrites, I'm kind of outlining them all here in red. My dendrites are the location of all of my chemically gated channels. Chemically gated channels. I only have chemically gated channels at places on my neuron where it's going to have a chance of bumping into chemicals. If I put chemically gated channels down here, there's never going to be a chemical there. That's a waste of a protein because the only place I'd ever have those chemicals is up here at the dendrites. So any kind of chemically gated channel is only ever found on the dendrites, up here on this receiving part of my neuron. The part of my neuron that processes all the different chemical messages that I received is this part right here of my neuron. What's this part of the neuron called? Where I do my processing of those messages. Yeah, so this part right here in my box, this part right here is called the axon hillock. The axon hillock. When I add together all of those messages, when I'm processing those messages, what is the name of that? Or in other words, what does the axon hillock do? What's its job? Yeah, a couple of us have chimed in. The job of the axon hillock is to do what's called summation, where I add together all the different messages that the dendrites received and figure out what they mean. The way that a neuron figures out what they mean is by seeing what charge we end up at. What's the name of that special charge that if a neuron gets there, it knows it's supposed to talk to its neighbor? What's that special charge called? Yeah, we're totally right. The special charge I've got to get to know that I'm supposed to talk to my neighbor is threshold. Threshold is the place where voltage-gated channels can open up. Everything up here that's going on with my dendrites, this is all chemically gated channels. When we start talking about voltage-gated channels is right here. I have to hit threshold here at the axon hillock for my voltage-gated channels to open up. In particular, which kind of voltage-gated channel opens to depolarize my membrane? Who makes my membrane charge go up? Yeah, so voltage-gated sodium channels depolarize the membrane. Depolarize the membrane. If I hit threshold at the axon hillock, voltage-gated sodium channels are going to open up. That's going to depolarize the membrane starting here at the axon hillock and all the way down across the axon, all the way down to these places down here called my axon terminals. An action potential, a moving change in charge, starts up here at the axon hillock, but it's got to go all the way down to the axon terminals for it to matter. So I have voltage-gated sodium channels all the way down, all the way here to my axon terminal. I'm going to find them everywhere. So we've got voltage-gated sodium channels depolarizing the membrane. Just as quickly, though, as I depolarize it and get it all the way up to positive 30, I need to get it back down to negative 70 again. Which kind of channel helps me to repolarize the membrane? Who helps me to repolarize the membrane? Yeah, now it's those voltage-gated potassium channels. Voltage-gated potassium channels are the ones that repolarize it, that help me get back to normal. So starting up here in the axon hillock, all of the places, let me draw it and I'll let go. All of the places that I depolarize the membrane, I'm going to have to repolarize it again too. So starting in the axon hillock, I use it to get me back to normal, all the way along the axon, all the way down to the axon terminals. Anywhere that you have voltage-gated sodium channels, you're definitely also going to have voltage-gated potassium channels. Because if I go up, I've got to come back down. So starting in the axon hillock, all the way down to those axon terminals, we're going to have voltage-gated potassium channels as well. My last kind of ion channel is what I find only at the axon terminal. Which specific kind of channels are only found at the axon terminal? What's left? Yeah, at the axon terminals, I have one last kind of voltage-gated channel. It's a voltage-gated calcium channel. Voltage-gated calcium channel. Let me move that. Voltage-gated calcium channels only found in the axon hill or the axon terminals, excuse me, axon terminals. Voltage-gated calcium channels don't depolarize the membrane. They don't repolarize the membrane. What's the function of a voltage-gated calcium channel? What do I use that calcium to do? What's its job? Yeah, so I like the way that we phrased it. Calcium helps me to talk to my neighbor by allowing me to spit out neurotransmitters. So here's a thought here with calcium, by the way. The only job of calcium is to help me spit out neurotransmitters. So the only place I'm going to have a calcium channel is the one place on my neuron where I spit out neurotransmitters. The only place on the neuron where you spit out neurotransmitters is down here at the end, at the axon terminals. So I have voltage-gated calcium channels here at the axon terminals. They bring calcium inside the neuron. when that calcium comes inside, neurotransmitters leave. Calcium and neurotransmitters, they don't play nice with each other. So calcium comes in, neurotransmitters come out. Voltage-gated calcium channels, the only place they're found is at the axon terminal. Now, the question that I got from multiple students was related to the review assignment, where it asked about the kinds of channels found at the axon terminal. We all know that we've got voltage-gated calcium channels there, because I use that calcium to spit out my neurotransmitters. But the hint that I gave my friends via email, and that I'm going to give to you guys as well, these are voltage-gated calcium channels. So for us to be able to get a voltage-gated calcium channel open, I have to have the types of channels that help me to depolarize and repolarize the membrane. So at the axon terminal, where I have voltage-gated calcium channels that help me to spit out my neurotransmitters, I also have to have voltage-gated sodium channels and voltage-gated potassium channels that are going to help me reach the voltage I need to open up the voltage-gated calcium channels. Question in the chat says, do chloride channels only occur in muscles? So muscle cells actually don't have chemically gated chloride channels or chemically gated potassium channels. The only kind of channels that muscles have are just the chemically gated sodium channels. The chloride and potassium channels are only seen on neurons. We don't see those channels on muscles. Muscles just has these chemically gated sodium channels on them. Any other neuron specific questions? Yeah, go for it, Miriam.
So I just need a clarification. So when we're talking about the voltage gated sodium channel is followed by the voltage gated potassium channel, that means that we have any part, if I can bring my pointer here,
Yeah.
any part. So when you highlighted like the blue part is as the sodium channel. So when the sodium leaves this part of the axon and moves, or the potential leaves this part to move to the next part, automatically the potassium gated channel opens, and so on till the end of the line. Is
Exactly.
that?
Yes, yeah. So if you look at the picture about the way that an action potential travels, it shows that first we bring sodium into that part of the axon. Here, let me see if I can find it really fast. First, I always bring in sodium to make my membrane charge positive, but immediately after I bring in that sodium, I need to spit out some potassium because I'm way too positive on the inside. So this is all one part of my axon. Sodium rushes inside, then potassium rushes out, which gets me back to my normal charge. Because the sodium rushed in right here, that makes this part of the membrane open up its voltage-gated potassium channel or sodium channels. But if I open up those voltage-gated sodium channels, I'm going to have to open up the voltage-gated potassium channels as well to spit that potassium back out. So each place that I have those voltage-gated sodium channels to bring in the sodium, that causes me to, in the next millisecond, spit out some potassium to balance it back out. So yes, they follow each other, essentially, is kind of a good way to think about it.
Okay. All right, thank you. And then the chloride channel is also at the end of the sorry, it's the axon terminals, right?
So When we talk about chloride channels, we only have chemically gated chloride channels. So there's only chemically gated chloride channels, no voltage gated chloride channels. So the only place I'm going to have chemically gated chloride channels is going to be the places where I have chemicals. And where did we say again that we have chemicals? Yeah, Tierney's right. The only place I'm going to have chemically gated chloride channels is on the dendrites. You might be referring to, here's a quick reminder for us. We don't have voltage-gated chloride channels. That's a distractor answer. There's no voltage-gated chloride channels. They don't exist. We also don't have chemically gated calcium channels. That's another distractor answer. It doesn't exist. So we've only got chemically gated chloride, sodium, and potassium channels. We've only got voltage-gated calcium, potassium, and sodium channels. So keep that in mind. We don't have chemically gated calcium. We don't have voltage-gated chloride. Look at the question here. Okay, so Ariel's asking about summation. Let me pull up our picture really fast and we'll look at that right here. The process of summation is when a neuron receives multiple messages and adds them together, allowing that neuron to figure out whether or not it's supposed to respond. When we're talking about the types of summation, there's two types, right? We have what's called temporal summation, and we have what's called spatial summation. When we talk about temporal summation, that temporal word means time. Temporal means time. Spatial means space. But the big thing when we were talking about summation that I told you to be familiar with is whether I'm listening to one neuron or multiple neurons. When we talk about summation, temporal summation, am I listening to one neuron or multiple neurons when I do temporal summation? Exactly. Temporal summation, listening to one neuron. Listening to one neuron. That one neuron is sending me 99 text messages. That one neuron's doing it. In spatial summation, I am listening to multiple neurons. They're all talking to me at different parts on my dendrites. I'm receiving 99 messages all over the place, like this neuron that we see right here. It's easy for us to think about, so Ariel was typing for us one of the questions from the assignment that asked about multiple excitatory or multiple inhibitory messages. It's easy to recognize that with spatial summation, we could hear a mix of types of messages, so a lot of excitatory messages and inhibitory messages. When we talk about temporal summation, we're going to hear a lot of different messages as well. We're listening to one neuron, that neuron is talking to us a lot. Anytime we're doing summation, we can always hear more than one message. So temporal summation and spatial summation, with both of them, we're hearing multiple messages, multiple excitatory or multiple inhibitory messages, or some kind of mix of those messages. Yeah, summation. And remind me again, what do we call this magic number? -55 right here. What's this magic called? Yeah, this magic number is threshold. So magic number threshold, if I hit threshold by doing summation, I'm going to go through and I'm going to talk to my neighbor. Yes, so to clarify with our types of messages here, when neurons talk to each other, each neuron can only send one kind of message. So this neuron right here that we're looking at would either always send an excitatory message or always send an inhibitory message to its neighbor. So if we're receiving excitatory messages and inhibitory messages, more than one type of message, that's going to be spatial summation. If we're hearing the same type of message over and over again, that's temporal summation. So temporal summation, always the same message, always the same message. Spatial summation, EPSP and IPSP. It can be a mix because more than one neuron is talking to me. But everybody always spits out one message, either an excitatory message or an inhibitory message. You might send me your message 10 times, or I might get 10 different messages. But in either type of summation, we do hear multiple messages. Thumbs up or other questions? So, yep, I'll mention refractory really fast. I want to do, let me see if I picked the right one here. We'll talk about, we'll use this to help us with refractory. So we're looking at the graph here. I 100% promise you that on your exam, I am going to ask you about the names of this part of the graph and which particular kinds of ion channels are open on parts of the graph. When we talk about the stuff that happens here below threshold, this place down here where I'm doing summation, which kind of channels are involved in my stuff that happens below threshold? Yeah, so everything that happens below threshold is because of my chemically gated channels. So chemically gated sodium, chemically gated potassium and chloride. The process of summation, where I add together all of the messages that I'm receiving, all of that has to do with chemicals. Once I hit threshold, I have hit a charge where now it's all about charge. Chemicals don't matter anymore. When you hit threshold, voltage takes over. So when I hit threshold, I open up voltage-gated sodium channels. Voltage-gated sodium channels. They open at threshold. They stay open this entire time here. So voltage-gated channels are involved in depolarizing the membrane. They first open here at threshold. Now, if I am at an axon terminal, when I make the axon terminals charge positive 30, I can open up my voltage-gated calcium channel. If I'm at an axon terminal. Because remember, calcium comes in, and that's going to allow me to spit out neurotransmitters. But if I'm still on the axon of a neuron, let's say I'm not at the axon terminal yet, I still do open up a type of channel when I'm at positive 30. What's the type of channel that I would open at positive 30 to help me repolarize the membrane? Who helps me to repolarize? Yeah, exactly. Voltage-gated potassium channels. They help me to repolarize the membrane. So I would open them up here at positive 30. They're going to stay open all down here as I repolarize the membrane. In fact, I hyperpolarize the membrane. I make it too negative because I spit out too much potassium. Over time, I'll get back to normal because of those leakage channels that slowly get me back to where I'm supposed to be. So voltage-gated potassium channels open at positive 30. they will stay open all through this process of repolarizing the membrane. Gloria has a question. Go for it.
So just to clarify, both of these, so this answer would be voltage-gated sodium, and so would this one. Is that correct?
Correct. Yes.
Okay.
This is where voltage-gated sodium channels open, and the type of channel that makes this process happen of depolarization are those voltage-gated sodium channels.
Okay.
So voltage-gated sodium channels first open here, stay open this whole time. Up here, voltage-gated potassium channels open, stay open this whole time over here.
Okay. Thank you.
We had the question earlier about what's called, let me type it up here in red, the refractory period. Refractory period. When we talk about the refractory period of a neuron, this is the time where a neuron either cannot do anything in response to another message, or that other message has to be like yelled at me for me to be able to do anything. There's two kinds of refractory period where it's hard for a neuron to hear things. So one of the times where it's hard for a neuron to hear things, actually it's impossible for a neuron to hear things, is the absolute refractory period. In the absolute refractory period, we can't hear a message at all. There's no way. The reason we have the absolute refractory period is because my voltage-gated sodium channels are tied up. We'll say they're tied up. They're busy responding to another message they already heard. Unfortunately, I didn't pull the picture, but can my friends help me out in the chat? That picture that shows the three shapes of the voltage-gated sodium channel, what page is that on in your notes? That shows the three shapes of that channel. Okay, so page 14, if you've got your notes in front of you, flip to page 14. It shows that there are three different shapes for the voltage-gated sodium channel. When it's waiting to receive a message, when we haven't quite got to threshold, it's closed in its channel. When we reach threshold, it opens up. While it's opened up, that's what's allowing a neuron to get ready to talk to its neighbor. When we get all the way up to positive 30 though, the voltage-gated sodium channel realizes, oh, we are way too positive. I'm not supposed to be open anymore. The channel itself stays open, but if you notice on the bottom of that protein, there's the little thing, I call it the ball and chain protein. It's kind of like a stopper in a bathtub. It folds into place and prevents any more sodium from coming inside. Until we get back down to about threshold, that stopper is in place and there's no way for me to reopen up that channel. I need to open this channel to allow me to respond to a new message. So if this channel is already open, because that's what's going on right up here, or if that channel is inactivated, because that's what's happening with it over here, if my channel is inactivated or it's already open, I absolutely can't hear you. There's no way. Once that channel is back in the right shape, so once the tub stopper is pulled out and stuff could drain down through that channel again, I enter what's called the relative refractory period. In the relative refractory period, my voltage-gated sodium channels are ready. They could respond, they could open up, but the problem is I'm actively making my membrane charge keep going down. I'm spitting out potassium. And remember, if I want this kind of channel to open up, it has to be at threshold. I've got to get to negative 55. If I'm repeatedly and actively spitting out potassium and dropping, we kind of gave the analogy, if you are skydiving and you need to get back up to your plane, it's going to be hard for you to get back up to the plane That's kind of what's going on in the relative refractory period. You're free falling, you've jumped out of the airplane, but we've got to get back up here for us to be able to open those channels again. If I have enough neurotransmitter that lets in enough other sodium, we could undo the effects of spitting out all that potassium, but it's going to be really hard. So in the relative refractory period, I could hear a message because my voltage-gated sodium channel is back in the right shape. It'll just be hard because my membrane charge is not playing nice. If we're in the absolute refractory period, I can't hear another message because my voltage-gated sodium channels are either already open, like we see right here, or they're inactivated. That's what we see over here. Absolute refractory period and relative refractory period. Okay, so Vanessa's mentioning a question on the review that says blank is a problem with potassium. Yes, so the relative refractory period, the reason we have that relative refractory period is because of potassium, correct? I'm spitting out so much potassium that my membrane charge is going the wrong direction, preventing me from being able to hear a message. So yes, we've got an issue with potassium in the relative refractory period. We've got an issue with my voltage-gated sodium channels in the absolute refractory period. Yeah, so Jacqueline asked the question, if we were to see on the exam, the question would be what opens up here? It would probably be two answers, although check to see if it asks you about the axon terminal or if it just asks you in general. The two kinds of channels that could open up here, the voltage-gated calcium channel at the axon terminal, the voltage-gated potassium channel everywhere. Always the voltage-gated potassium channel will open at positive 30. At the axon terminals, the voltage-gated calcium channel will open as well. All right, we are unfortunately out of time. We hit everything except the homunculi. So I'm so sorry, Jacqueline. I know. We did talk about that. Well, here, we can do a really quick crash course. Yeah, I will be here at 5 too. So if we have more questions, we can come back at 5. Here's my crash course, and I'll refer you back to those videos because we did talk about them in class together. This is in the post-central gyrus. So this is the bump that's behind the central sulcus. This is the one where we have what's called the homunculus. This is a map for you of where in your brain we send sensory information from each part of your body. So the texture that you feel of your pencil would be sent to this place on that bump. The texture of the socks that you're wearing would be sent right here. If you walk outside and it's cold, you feel cold air blowing on your face, this is where it would be sent. On the sensory homunculus, we get an idea of where in the post-central gyrus the general sensory information goes to for your brain to figure out what you're feeling or what the temperature is or that kind of stuff. A quick reminder that we have what's called general senses, and then we have what's called special senses. General senses, things like touch, temperature, texture, pain, pressure, I'll add that to the list. These are the kinds of things that we would send to somewhere in the post-central gyrus. The special senses were things like vision, like hearing or taste and smell. These kinds of things, special senses, don't go to the sensory homunculus. They're going to go to other places in the brain. The places that we studied in our packet places like the occipital lobe or like the temporal lobe. So general sensory information, these kinds of things from various places in your body, this is where they go in the brain. If I have a really big place on this sensory homunculus, like the fingers, for example, or like the foot, really big place here, what does that tell me about those parts of the body when a region is really big? What does that tell me? Yeah, exactly. It tells me that I can receive a lot of sensory information from those places. So the bigger a body part on the homunculus, on the map, the more information I can receive, the more sensory neurons that live there. Yeah, like Gloria said. Contrast that with the motor homunculus. The motor homunculus is the one from our assignment here at the beginning of class that we talked about in the pre-central gyrus. The pre-central gyrus is part of the frontal lobe. Now we're talking about movement. The bigger a body part is on the motor homunculus, the more motor neurons that live in the frontal lobe that control it. So something like the hand or the fingers that are very large on this homunculus, lots of different motor neurons send information to those fingers to allow them to do lots of different kinds of activities. Or lots of different neurons control the lips. There's lots of motor neurons in the lips that could go to that part of the body. I can send lots of different directions to that place. Now, your forearm and your arm, not nearly as many motor neurons that go to those places. They won't be able to do as many different things. So the size of body part on the motor homunculus gives us kind of a general idea of how many different kinds of information I can send to those places. So I'm looking at Nicole's question. Yeah, so technically we would say that we can do fewer actions with the tongue than we could with the hands, or kind of with the fingers in general, just because their sizes are different. Your tongue, you can do way more with your tongue than you can with your forearm, for example, or about as much with your tongue. This little tiny tongue receives just as many sets of instructions as your entire forearm. So the things that control the fingers, Nicole, the muscles that control the fingers are actually found in this hand region. So when we talk about moving the fingers, they kind of work one and the same. We don't actually have a lot of muscles that are specifically just in the fingers. They're mostly in the hand controlling the fingers. Yeah. All right. I know I didn't spend as much time on that as we would have hoped for, but I think I've got a hungry baby who is waiting for me. So I may have to call it quits for this morning. But I will be back at 5 o'clock, so if multiple of you show up, we can definitely talk about stuff. Or if you just have 1 or 2 questions, you want to show up by yourself, I will be here. So 5 to 6:30 tonight, please feel free to stop by. Jacqueline about to take the exam. Well, you can always try to chat it up with some of your classmates in the group me if there's particular questions you have left. But I think we're gonna call it quits for today. Good luck on the exam to all my friends here who are taking it and to all my friends on the recording. I'm going to go ahead and stop our recording. I'll stick around for just a couple minutes in case we have any last minute questions, but then I'm probably going to have to peace out.